Hi again, everybody. It's Bill Cavanaugh from abscopalevents.com, where we are going to keep talking about the abscopal effect and, possibly, the emergence of a whole new way to look at cancer treatment. In case you're new here, the abscopal effect is the observation of an untreated cancer, typically a metastatic deposit of cancer, regressing or shrinking following the treatment of a targeted and probably related cancer, often the primary cancer. So, one cancer is treated, another untreated cancer shrinks. Abscopal means away from the target. Once considered the rarely, if ever seen, unicorn of cancer treatment, the abscopal effect has been talked about much more frequently in the biomedical literature of late. While no one can yet make the abscopal effect occur on demand, the point we're trying to make here is that the abscopal effect is almost certainly the work of the human immune system. And our hypothesis is that the increasing mention of the abscopal effect relates to the widespread deployment of the first generation of immunotherapy drugs known as immune checkpoint inhibitors that were first introduced about 15 years ago. We're just going to call those immunotherapies here, FYI. And we're going to keep watching the literature and watch as this phenomenon grows. And of course, we will keep wondering why more significant, concerted clinical research is not going on. We are archiving earlier episodes here in case you're interested. We're building a narrative, if you will. Please subscribe or like. There's more coming. Now, before we go through another actual abscopal event, I want to finally get to talk about a recently emerging and truly fascinating idea that might change a lot in how we look at both cancer and cancer immunology, and maybe even more than that. If you recall a couple of episodes ago, we talked about cancer cells with their twisted, contorted appearance and invasive behavior, and how it is postulated that mutations in DNA can lead to defective proteins that may contribute to what we call the malignant characteristics of cancer. Since it is so fundamental to getting a handle on what cancer actually is and what may be happening in terms of where cancer comes from, let's illustrate this idea again. Starting down at the cellular level, here you can see a cartoon example of hypothetical prostate gland tissue under the microscope. It's just a cartoon, of course, okay, not real. Prostate, and being a good example of an epithelial tissue that often transforms into carcinoma or cancer. Now, this is an AI depiction. I think it's pretty good, actually. And given it's a cartoon, we are leaving out some detail of the nucleus and some other things because we want to focus on the loss of architecture as epithelial cells transform into carcinomas. On the left, we have schematic normal prostate tissue. I think you can appreciate the really beautiful multi-layer architecture of these benign glands. They are all isolated from one another and look like lily pads floating on a lake of what is called stroma, which is the in-between milieu or supportive matrix of the organ. It might help to see that we are cutting through glands, which are secretory blobs that express fluid into tube-like structures called lumina. So what we see is a two-dimensional image of basically a three-dimensional cluster of grapes, Now, on the right, we have a schematic illustration of prostate cancer. On the left side of that image, you will see some well-differentiated low-grade cancer. The glands still look like glands, but they have lost that multi-layered structure, and you can see some of them starting to fuse together, which they're not supposed to do. And on the right of that image, you can see poorly differentiated or high-grade cancer in all solid purple just invading into the frame. That's a scary-looking prostate cancer. These cells have stopped organizing themselves as glands And along with the obviously different look, we get what I think you can kind of appreciate as an aggressive and invasive crowd of cells. These poorly differentiated cells are the type that become metastatic as well. So, how do our well behaved epithelial cells get from forming these nicely organized and useful structures into this sheer disorganization of invasive cancer? If you recall from our earlier episode, as we went through our talk about biology, it is generally thought that, on one hand, mutated or corrupt genes made up of DNA end up making cancer allowing or cancer assisting proteins. And these proteins, or gene products, contribute to the look and behavior of cancerous cells. Let's look at it again. Here's another very generalized illustration that we've talked about before. In the top figure, we see the normal process that occurs in all epithelial cells as they divide again and again, with normal genes being translated into normal protein that supports a normal cell. On the bottom, we see a hypothetical mutation in a gene that results in a defective gene product, or protein, that aids in the transformation of a normally functioning cell into a carcinoma cell with all its malignant features. Again, this is very generalized, but also very consistent with how cancer is viewed biologically. The second general idea is that there is an overpresence or overexpression of certain genes that result in more copies of certain proteins being made than there are supposed to be. Or proteins that aren't supposed to be made at all. For instance, beyond very early developments, alpha-fetoprotein, for example. But in all these cases, the mutated and or amplified and or not supposed to be activated anymore genes that drive the science of cancer biology all begin or have their source as a part of the regular human genome. In other words, according to standard thinking, cancer is believed to arise from badly performing genes that are encoded in the human genome. Now, please bear with me. I promise that this is going to get more interesting. Now, in order to understand this regular human genome idea, let's step back just a bit. Let's go back to 1987 when I was getting my degree in biology. At the time, the scientific world was lit up with news that soon there would be something called the Human Genome Project, and that DNA sequencing technology was improving vastly and would be used in this effort. The Human Genome Project was going to identify all the genes that make up the human genetic repertoire. And at the time, optimism was very high. I remember legitimate talk about human disease being conquered, cancer figured out, and on and on. It was going to be a very big deal. Now, genetics as an idea had been around for a while. Gregor Mendel was inventing genetics as a science back in the 1800s, and the idea of hereditary through generations was pretty well acknowledged. Everyone had already speculated that genes were the unit of heredity and that DNA contained these genes or this genetic information. The building blocks of DNA had been identified in the 1920s, and Watson and Crick figured out the double helix in the 1950s. So, we have the idea of heredity, the idea of genes, we have the DNA building blocks, and the backbone or the construct of the DNA molecule itself. But nobody really knew what was in there until the painstaking work of sequencing began. And getting back to our index year of 1987 and into the 1990s, nobody actually knew how many genes were in the human genome. Now, DNA sequencing had been going on for years, but it was very slow and only made sense for subcellular organisms like viruses. And by the end of the 1970s, the genomes of the first of the DNA viruses, the smallest things imaginable, which aren't even considered genuine life, were sequenced. But it was something. You can see here a base pair referring to one bridge of information between the two DNA helices, the base pair being the most basic unit of the DNA molecule. This DNA virus, or bacteriophage in this case, had a tiny genome of 10,000 or so base pairs and a whopping dozen or so genes. Now, at the time, this was huge progress. It was well appreciated that viruses would contain just a few genes, being that viruses can't really do anything on their own. They exist by parasitizing real cells. But the debate was sparked as to just how many genes would be contained in the human genome. There was great speculation, I remember well, in the late 80s and 90s as to just how many human genes there were, how many genes it took to make a human being. It was well known for decades that each somatic human cell nucleus held 23 pairs of chromosomes. The virus had no such payload, so the human genome was going to be vastly greater than the genome of a virus. And guesses ranged, I remember this well, from 100,000 to 1,000,000 genes in the human genome. It would have had to be, the complication of humans would have to have been vast. Then, in 1998, the first big genome was sequenced. It was this little critter, Caenorhabditis elegans. We'll just say C. elegans, the elegant roundworm as it was known, a tiny little nematode worm. A whole 0.04 inches long, this roundworm, just at the edge of visible without a microscope, if you have really good vision. But it was a free-living animal as far as biology was concerned. And lo and behold, its genome was 10,000 times the size of the virus's and encoded a whole 20,000 genes. So those guesses of at least 100,000 genes, and maybe 1,000,000, in the human genome seemed right on target. And right around 2001, 3 years after C. elegans was sequenced, we all got the answer at long last, thanks to the high-speed sequencing networks that came online, care of the Human Genome Project. And humans ended up with 3 billion base pairs, 30 times in terms of base pairs, 30 times more than our little worm friend. So, in this corner, we have a barely visible hermaphrodite worm with its 20,000 genes, right? And in the other corner, we have the species responsible for skyscrapers, handheld computing devices, space travel. And guess what? Also 20,000 genes. So it turned out that we and our worm friends were built from genomes that contained pretty much the same number of genes. I remember that being quite the shock at the time, and I'm sure to a lot of people it's still shocking. But what does this all mean to our conversation about cancer and the abscopal effect? It means a lot, and I'll tell you why. Because despite what everyone might think, it can be persuasively argued at this point that the human genome, by which I mean the entirety of the fully identified set of human genes, 20,000, is not what is driving things biologically. And for everyone who thinks that there is a gene for smart, for tall, for attractive, or for white, for black, athletic, not athletic, and so on. There is not. There's no such gene for any of these things. Now, here's a table containing the main categories of the 20,000 genes in the human genome. It turns out that the genome contains an instruction set and toolbox for building cells and keeping them functioning, and, importantly, for building systems that allow for cells to communicate with each other, both nearby and far away. And here's a table containing the main divisions of the 20,000 genes in the Caenorhabditis elegans genome, the nematode worm. These are surprisingly similar function-wise, okay? And we realize how vastly different the two organisms are, okay? Remember that, okay, for our purposes, all these genes encode proteins, which create and do the business of cells. And that's what genes are, a highly advanced system for building and maintaining cells, which are the unit of life itself. Now, we need to break this conversation in half because we're running long. But I will be back very quickly to tell you the other half of this story and how it relates to cancer, human cancer, and possibly to the abscopal effect. Just please remember that our current mainstream ideas of cancer development and the subject of many, many treatments under development work from the understanding that genes from our genome, the 20,000, undergo mutation and result in proteins that aren't right and that contribute to the development of cancer. But here's the thing. Of all the base pairs in the genome, the 20,000 genes only take up 2% of that informational space. In other words, of all the information located in your chromosomes, in those 3 billion base pairs, 2% are codable genes representing the whole of the entire genome. And 98% is something else. It's that something else that we're going to talk about next. Spoiler alerts, that 98% may be running very well, running the whole show. Hope to see you next time. Signing off for now.